Hi, I'm Michael Pointer, and you're listening to Abstract Bioscience, a podcast about new and exciting research in biological sciences taking place across the Norwich Research Park and beyond. Today, we're looking at recombination, the way that parental genes are mixed when producing sex cells. But before we meet today's guests, I think the background is going to need some unpacking. Let's start from the beginning. If individuals within a population differ from each other, some, with more favourable characteristics, are able to survive longer and leave more offspring than those less well suited to the environment. This is the reason that genetic variation is one of the fundamental requirements for evolution to happen. One well-known way in which variation is generated is through mutation, but another important source of variation is genetic recombination. Recombination takes place during meiosis, a special type of cell division that generates sex cells, or gametes. In animals, these are the sperm and the eggs. In all cells, DNA is arranged into units called chromosomes. Many animals and plants we call diploid because they have two copies of each chromosome, one that they got from their mother and one from their father. Sex cells, though, are haploid, having only one copy of each chromosome, and so contain only half the genetic information required to produce a new organism. So that means when two gametes come together, they combine to form a diploid zygote. So now we know that gametes only have one chromosome copy, but here's the clever part. That chromosome is not just the same as either the maternal or the paternal chromosome. Each sex cell gets a combination of the two. In other words, the process of recombination creates a unique mix of maternal and paternal genes to go into every sperm or every egg. This is why full siblings are genetically different, unless, of course, they're identical twins. Are you with me so far? Good. Recombination swaps genes between paired chromosomes, and it does this by creating a lot of breaks in the DNA at the same positions in the maternal and paternal chromosomes. Then, during the repair of some of these breaks, the broken end is not joined back to the same chromosome, but to its partner, and the two swap a section of their DNA. This is known as crossing over, and it's a much easier concept to explain with a diagram, so quickly ask Google if you're struggling to picture it. The amount of crossing over that happens during recombination is very important. Too few crossovers doesn't create enough variation, but too many risks breaking up beneficial combinations of genes. The number of crossovers and where they form is regulated by crossover interference, although the exact mechanism isn't well understood. Today's guests, Chris Morgan and John Fozard from the John Innes Centre, have a new paper out in Nature Communications focusing in on the intricacies of meiosis. Welcome to you both. Could you tell us who you are and what you do?
Hello, I am Chris Morgan. I'm a BBSRC Discovery Fellow at the John Innes Centre, and I guess I primarily use fancy microscopy to try and understand how meiosis works in plants.
Hi, I'm John. I'm a mathematician at the John Innes Centre. And I guess my thing is using a combination of image analysis and mathematical modelling to try and work out what's going on in biological systems.
Thanks for joining me today. I'm looking forward to getting into this. So, you have this great new paper together, which broadly is about this phenomenon of crossover interference. So, as a starting point, could you first define what crossover interference is for us?
So, I guess I'll start with the history of crossover interference. So, crossover interference was first identified over 100 years ago in some of the earliest genetic linkage experiments in fruit flies in a famous lab in Columbia State University. And they found that during meiosis, when you get a crossover occurring at one position along a chromosome, it inhibits the formation of crossovers nearby. And so the result of that is that crossover interference limits the total number of crossovers that occur along chromosomes during meiosis, and also makes sure that crossovers are reasonably evenly distributed across the length of a chromosome.
I see. So this happens across almost all sexually reproducing organisms, but you work with the model plant Arabidopsis thaliana. Why is that a good model for studying recombination?
I mean, a lot of the fundamental science investigating genetics and meiosis has been performed in plants over the years. And the obvious example that sort of springs to mind is Gregor Mendel and his peas, well over a century ago. And one of the reasons that people like using plants for looking at meiosis is that a lot of plants have very big chromosomes, which makes looking at them down a microscope very easy. Now, Arabidopsis is actually the opposite to that. It has very, very small chromosomes. But what that means is that it's very easy to sequence the genome of Arabidopsis. And it's also very easy to do genetic experiments in Arabidopsis where you mutate a particular gene and then see what happens, basically. And so the kind of tractability of Arabidopsis to these genetic experiments means that we've been able to investigate meiosis in very high detail in Arabidopsis and to kind of determine which genes are responsible for which particular parts of meiosis. So we've identified a large number of the genes, for example, that are involved in crossover formation. And so because we know a lot about meiosis and Arabidopsis because of these genetic resources that are associated with it, it's a great system for studying meiosis in plants.
So you're looking at Arabidopsis cells undergoing meiosis, and you focus on this molecule called HEI10. What was known already about its role in crossover interference?
So we tend to call it high 10, just because it rolls off the tongue a bit more easily. And so a reasonable amount was known about high 10 before this study. So originally a group in France, Mathilde Grelon's group, first characterized the mutant of high 10 in Arabidopsis and showed that it was essential for crossover formation. If you knock out high 10, you don't get crossovers. And then following on from that, Ian Henderson's group in Cambridge, who's actually a co-author on this paper, he used some population genetics approaches to show that when you vary the amount of high 10 in a cell, you actually change the frequency of crossovers. So if you increase the amount of high 10, you get more crossovers. If you decrease the amount of high 10, you get fewer crossovers. And so quite a lot was known about high 10 in Arabidopsis already, but also because meiosis is a very conserved process. So by that, I mean that meiosis in plants is very similar to meiosis in animals and very similar to meiosis in fungi as well. Because meiosis is such a conserved process, we can also look at the versions of high 10 in other organisms. And so we can look at those other studies and then infer from those proteins perhaps how high 10 operates in Arabidopsis.
Okay, that's a good jumping off point, but still plenty to discover. So then, what were the questions that you set out to answer?
The idea is that Chris came to us with these observations about high 10. He could see that there were early during this stage of meiosis, there are a lot of small dots of high 10 on each of the chromosomes. Whereas later during meiosis, these reduce down to maybe being, 2-3 dots of high 10 on each chromosome. And I think the idea was that we wanted to explain whether a simple model where high 10 is able to move around on the chromosomes and kind of aggregate at various points, which in the paper we call recombination intermediates. So these are probably the double strand breaks that happen in meiosis. So the idea is high 10 is able to move between these recombination intermediates. And then over time, a small number of these recombination intermediates are selected to become crossovers. So can a model where the high 10 moves around and aggregates at these RIs, can that explain what's going on? And then the idea came about that, maybe we could work together and make a model for this process to see if such a simple model is actually able to explain what's going on. So can you get crossover interference in such a model? Will it stop nearby, will nearby DSBs be less likely to form crossovers? And also, can you explain the patterns of crossovers along each of the chromosomes. Basically, sort of when you have a single crossover, it tends to be found near the center of a chromosome, whereas double or triple crossovers tend to be found more towards the ends of the chromosomes. So can the model recapitulate that patterning? Chris came up with this idea, and he basically made a very prototype version of the model in Excel. So we took that very simple prototype and we developed it into a more complicated mathematical model. There are some equations in the paper, which we then could simulate and compare with the experimental data.
Great. But before we really get stuck into how you went about doing that, I think we need to define what a coarsening process is, because it's really key to understanding what your model is doing. John, I think that might be one for you.
I can have a go. So this model is basically a coarsening type model. So people use these kind of models in physics all the time. They're often applied to things that are mixtures of two different phases. For instance, emulsions of oil in water or droplets in clouds. And the third kind of typical thing is soap bubbles. And in this kind of model, what happens is basically the objects, so the raindrops in the cloud, which are bigger, tend to grow at the expense of the objects that are smaller. And often this is because it's easier for the water to escape from the smaller raindrops than it is to escape from the bigger raindrops, which means that because the water vapor can move around in the cloud, or for instance in the oil-water emulsion, the oil can kind of diffuse around in the water, this means that the bigger things grow at the expense of the smaller things. And you find that over time, the big things grow, the small things decay, and so you're left with just a small number of large things at the end. So that's the overall framework of the model, and they've been studied by quite a few different people in different contexts.
It's actually quite intuitive when you start thinking about water droplets engulfing other ones on a window or something. But maybe not something that would normally scream maths to me. Maybe I'll think differently next time it rains. As we've mentioned a couple of times already, this project came out of some microscopy work that you were doing, Chris. Would you explain what you saw there in a bit more detail?
Yeah, so originally I was trying to actually map recombination sites cytologically, and I was using HEI10, protein in this paper, as essentially a marker that marked crossover sites along chromosomes, and then you could image those chromosomes and then mark physically along each chromosome where a crossover site is. And then whilst I was doing that, I was actually working in another plant called Arabidopsis aeruginosa at the time, which is related to thaliana. In aeruginosa, most chromosomes only have one crossover, and occasionally they have two. And I noticed when I was trying to map these sites using HEI10 that when you got two HEI10 foci on one chromosome, they tended to be dimmer than when you got one HEI10 focus. And then when you combined that observation with some of the other stuff that was known about HEI10, like the fact that if you increase the amount of HEI10, you get more crossovers, you decrease the amount of HEI10, you get fewer crossovers, this kind of led to this idea of potentially there might be this coarsening mechanism of HEI10 that was driving crossover interference. And crossover interference really is something that I've always found very, very fascinating, because it's kind of one of the great remaining mysteries of genetics. It's been known about for a very long time, and lots of elegant and theoretical models have been proposed to explain how interference might work. But so far, most of them have really lacked any kind of robust experimental evidence. And so kind of these observations, I guess, led to us trying to develop a new model that we thought would sufficiently be able to explain crossover patterning, not just in Arabidopsis, which we have shown, but also I think would be able to function more broadly across evolution in different organisms and explain crossover patterning in pretty much all sexually reproducing eukaryotes.
Cool. So in order to do that, you adopted a modelling approach. Why was that the way you chose to go?
So I think the reason that we adopted a modelling approach is simply because trying to image this coarsening process in real time was essentially unfeasible. just due to the experimental limitations of trying to do that. So all the imaging we were doing was looking at fixed cells. So we're looking at static snapshots of meiosis in a cell, but we weren't looking at these dynamics in real time, and we weren't doing live cell imaging, which people have started to do in Arabidopsis meiosis, but it's a fairly new technique, and it's difficult. So we use a fixative called paraformaldehyde that basically puts everything into suspended animation. So everything stays the way it was when you made that slide. And so then we can image those cells, but when we're imaging them, we're just capturing one particular point in time. And so they're not alive anymore. And so then once we captured lots of these images of cells at lots of different time points, we could then compare that against the model to see whether this model, which was, actually did have dynamic simulations that mimic this coarsening process, to see whether the kind of quantitative measurements we got from the model matched up with these quantitative measurements that we could get from these static images. John, do you have anything to add to that from the modeling front?
I think we felt we had to make a model to understand whether we had enough of the components of what was going on in order to explain the behavior of the system. So, for instance, there were lots of questions about whether there should actually be other things happening at the same time, whether there should be other proteins involved, whether there should be other kinds of processes going on. Often, for instance, people think about patterning in biological systems in terms of a Turing model. So these have been made famous because Alan Turing came up with them, I think in the 1940s. And they've been used to explain many things such as stripes on zebrafishes, and they're very common in development. But a Turing system generally involves two chemicals that can diffuse. And by having this combination of these two proteins working in tandem, you can get patterns like stripes and spots and other things. But we wanted to know whether just this very simple model with a single protein involved could actually explain what was happening. And also, by using the model, we could make predictions. So we developed the model just using the wild-type data. So we just looked at what was going on in a normal Arabidopsis cell. However, we wanted to know what the effect of overexpressing this HEI10 protein, so having more of it, or having less of this HEI10 protein would be. And we could make predictions with the model for these two cases and then compare them with the data. And this gave us more confidence that actually we understood the underlying mechanism.
Okay, I see. It seems like a modelling approach was really useful to solve the kind of problem that you had here. And as I guess we've been heavily implying all through this discussion, you found that your model was very good at reproducing your experimental observations. So then, what does the way that the model was put together tell you about the way HEI10 mediates crossover interference?
Yeah, exactly. So you load a set amount of HEI10 onto your chromosome at the very beginning of meiosis. And to form a crossover, you have to start with a DNA double-strand break. And during meiosis, you get an excess of double-strand breaks. So in Arabidopsis, you get about 150 double-strand breaks, but you only get about 10 crossovers. So in our model, we load HEI10 at the sites of double-strand breaks, although we call them recombination intermediates, because at the stage that you're loading HEI10, they'll be partially repaired, these double-strand breaks, so they'll be recombination intermediates. So we loaded HEI10 at lots of these sites, at these recombination intermediate sites, as well as along the chromosome between those sites. And then the HEI10 can diffuse along the chromosome between these sites. And then, as John said, due to this coarsening process, some of those HEI10 foci at recombination intermediates will get slightly bigger, and the bigger ones will just naturally outcompete the smaller ones for the sort of limited availability of this HEI10 protein. So that essentially the big foci will suck up all the HEI10 from the surrounding area. And it's because they have this kind of sucking ability to suck up the nearby HEI10. That's what actually causes interference, because you deplete HEI10 within the kind of nearby vicinity along the same chromosome. So if you've got a high-10 focus at the other end of the chromosome, that's going to be less affected by this sucking process of this focus compared with one that's nearby. And so that kind of naturally explains this phenomena of crossover interference.
And then how does that relate to chromosome length?
So it's been known for a long time within meiosis research that longer chromosomes, so physically longer chromosomes, have more crossovers. And within our model, the reason for that is that on longer chromosomes, we're loading more high-10. And that actually fits with what we see using the microscopy, that the longer chromosomes seem to have more high-10 on them. And so because longer chromosomes have more space for larger foci to form without interfering with other foci that are slightly further away, that's why on longer chromosomes you tend to get more crossovers, because this interference essentially gets weaker the further away you get from one of these large foci.
Yeah, nice. I think I get it.
Phew. Job done.
This mechanism then that you've uncovered, elucidated, what implications does it have for recombination in Arabidopsis?
So I guess in terms of Arabidopsis, as John said earlier, we can make predictions using the model. And so we can kind of predict, if we increase high-10 by a certain amount, we can predict what effect that is going to have on recombination frequency. Now, in Arabidopsis, that's not particularly useful, because Arabidopsis, aside from being a great experimental system, isn't that useful in the real world. But, of course, by learning about recombination in Arabidopsis, and particularly because meiosis is such a conserved process, this can tell us a lot about how recombination is operating in more economically important plant species, so things like crops. And in a number of crops, for example, wheat, crossovers tend to actually be restricted to certain regions of the chromosome. Crossovers in wheat often tend to be located towards the end of a chromosome. And there's actually a large portion of the wheat genome that is kind of silenced for crossovers. And what this means is that there's a large portion of the wheat genome that is unavailable to wheat breeders to introduce new genetic combinations into their wheat breeding programs. And so by finding a way of modifying the recombination or crossover landscape within plants, then this may help to unlock this full genetic potential of wheat and make it available to crop breeders to hopefully be able to develop better varieties of wheat with desirable traits.
That sounds fantastic. Manipulating crossover positioning to create brand new genotypes. You talked a bit there about uses in other crop plants, but how well do you think it might generalise more broadly across the tree of life?
I think this coarsening process could potentially explain crossover patterning in most sexually reproducing eukaryotes. There are some organisms that actually don't exhibit interference at all, but they're in the minority. And they actually don't, most of those don't have high-10. So that gives you a clue as to potentially that this model may be at least vaguely correct. So given the conserved nature of high-10 in different organisms, and the fact that most organisms have a version of high-10 that does something similar and also has similar dynamics, so people have looked at the behaviour of these other versions of high-10 in different organisms, and they all tend to have this behaviour where they get loaded along chromosomes, initially seemingly along the entire length of the chromosomes, and then they sort of coarsen down to having a few foci located on chromosomes that then mark the position of crossover sites. I think given that that is such a conserved feature of meiosis, I think that this coarsening model certainly should be able to explain crossover positioning in a pretty wide variety of different organisms.
We probably do have to do some more work to look at these other organisms, and there are some big differences. Like chromosomes have very varying lengths between different organisms. In yeast, they're very small. Human chromosomes are at least very long genetically, but not quite as long as you might expect in terms of physical length. And these differences in length scale and also duration of meiosis mean that perhaps there might be additional players or additional processes controlling how fast the whole thing happens in these different species. So we're going to have to look a little bit more. We don't think that just taking the model and applying it to the other species directly will necessarily work. There may well be other differences, for instance, in terms of end corrections or how we deal with the different chromosome arms. We think there's potentially more complexity as you look at other organisms, but the basic idea, however, should apply in all cases.
Wonderful. Well, I think it's a really great paper and a great advert for collaboration. The way that the two of you work with very different methodologies, but they've come together so well in this particular application.
Yeah, I think it's definitely been a true interdisciplinary research collaboration where the mathematical modelling is an essential part of the paper and the kind of microscopy is an essential part of the paper. And when you combine them, I think they produce an even greater result than in isolation. And they really help us to get to the heart of the mechanism that drives crossover interference in a way that, I don't think we would necessarily have been able to if we were just, for example, from my perspective, if I was just using kind of experimental microscopic approaches, I don't think I would have been able to uncover the mechanism in the same way that we have with the addition of the modeling.
Yeah, I think the challenge often is that you have to learn a lot of new stuff. It's very difficult to do that in the time frame of a project. So as a modeler, you tend to work on different things. You can't really just work on one thing. You need to be moving from project to project to get exposure to new ideas. And what that means is that there was no way during the course of this project that I was ever going to learn everything about meiosis. I know more than I did when I started, but I don't know that much. And the important thing was to actually have good conversations with Chris, who is an expert in meiosis, who could help us guide the model and tell us what the important things were to include in the model and what the important things were to leave out. And it's that dialogue that you need to have that actually is the most important thing for making these kind of collaborations work.
And speaking of collaboration, I know it wasn't just you two, so do you want to give a nod to your co-authors?
Okay, so on the paper, there's Matthew Hartley, who helped us out with the image analysis. He developed some of the initial methods for measuring the HEI10 levels along the chromosomes. There's Ian Henderson, who helped us out by giving us the HEI10 mutants, so the under and overexpressors. Kirsten Bomboys, who was Chris's supervisor at the start of this project. And Martin Howard, who was my boss and who's a modeler, and he basically designed the initial model and had the initial discussions with Chris, along with helping a lot with the development of the project.
That was Chris Morgan and John Fozard discussing their new study, Diffusion-Mediated HEI10 Coarsening Can Explain Meiotic Crossover Positioning in Arabidopsis. I'll put a link to the paper in the show notes, along with a couple of other relevant reading items for the enthusiastic among you. As Chris mentioned earlier, crossover interference has been a bit of a black box within evolutionary biology, known about for a long time but not well understood. As research becomes increasingly interdisciplinary and collaborative, scientists can approach old problems from new angles and see things that were previously hidden. I think this is a great strength of the Norwich Research Park, where a cluster of institutes and institutions with different focuses allow researchers with different specialisms to rub shoulders, and it would be shocking if some excellent science didn't come out of it. I think that just about wraps up this episode. Thanks again to Chris and John. I'll be back with more science for you in the next episode of Abstract Bioscience very soon. In the meantime, show notes, details about the podcast, social media accounts, and links to information about the Norwich Research Park can be found on the Abstract Bioscience website at abstractbioscience.co.uk. The show is written and presented by me, Michael Pointer, and you can contact me at abstract.bioscience.podcast@gmail.com. We're on Twitter and Instagram, and our excellent theme music is by Phoebe Troop. Thanks for listening. Catch you next time.